going to introduce our next speaker, who's Dr. Husseini Manji. Husseini is the global head of Johnson & Johnson's Science for Minds, which actually Science for Minds is a new program, which I think he'll tell us a little bit about. He was previously the global therapeutic head for neuroscience at Janssen. Janssen's the pharmaceutical division of J&J. Before joining J&J, Dr. Manji was the chief of the Laboratory of Molecular Pathophysiology at the NIMH in the intramural program, and director of the NIH Mood and Anxiety Disorders program, which is, I think, the largest program of its kind in the world. His research has generally helped us to conceptualize neuropsychiatric disorders as genetically influenced disorders of synapses and circuits. And much of that work has prompted the investigation of new drugs and new therapeutics for patients who aren't responding to the current group of compounds. As an example, his work led to the FDA, EC, Canada approval of the first novel antidepressant mechanism in decades, as Ketamine, which has the J&J name of Spravato, which is a nasal spray for adults with treatment-resistant major depressive disorder. Dr. Manji has received a ton of prestigious awards. I'm not going to list them all, but you should know he's a visiting professor at Duke. He's an honorary fellow at Oxford. He's a member of the World Dementia Council, a member of the Scientific Advisory Board of the Stanley Center at the Broad Institute at MIT and Harvard, and a member of the World Economic Forum, their Global Futures Council. On a more personal note, Husseini, I think you're the only person on the program today from Big Pharma. And that's important, I think. During this pandemic, all eyes have turned to pharma for therapeutics and vaccines, including the announcement this morning from Pfizer about their first vaccine results. But let's face it, there are a lot of people who distrust pharma and talk about it as the evil empire or the dark side. You know the terms better than I do. I think it's important for people to know that in your years at J&J, you've shown us really what pharma leadership can look like. Your commitment to global health, your commitment to anti-stigma campaigns, your writing about how we have to think beyond the pill. For me, it's much more Luke Skywalker than Darth Vader. In your new role in the Science of Minds initiative, as I get it, once again, you're kind of looking at how do you bring in now the voice for what patients need, looking for ways to revolutionize care. Your leadership, your focus on improving care and improving outcomes should remind all of us that just as with the battle with COVID-19, the solutions of our mental health crisis will require both the private and the public effort, and hopefully a public-private partnership. So it's great to have you here today. We're really looking forward to your comments. Thanks for joining us.
Thank you, Tom, for that very, very kind introduction. Thank you, Kunal, Pushkar, Inscopix. It was such a timely event. And as Tom mentioned, and I think it's really coming back to the beginning as Brandon kicked it off with such a spectacular presentation, I think it's reasons to be optimistic and have hope. As Tom alluded to, that working together, we can make a big difference here. And it's not just about medications, it's about thinking about things much more holistically and coming together to make a difference. And certainly the COVID-19 pandemic has shown a spotlight on, where we really need to focus our efforts on. Hopefully what I'll try and do is talk a little bit about why I think there's reasons to be optimistic. One of the things Tom indirectly alluded to is that in the pharmaceutical industry, although the unmet need is so great, a lot of companies have actually pulled away from neuroscience in general, but even more so psychiatry. And why would that be? And I think this slide tends to capture why that's happened, is that companies have really taken the view that these psychiatric conditions are just so complex, and we just don't know enough about them. If we knew enough about them and what to target, we could make a difference. But right now, we just don't know enough about them. So let's go and invest in oncology and immunology. And I'm not saying that they shouldn't do that, and that when the science is further along, we'll come back here. I think as you'll hopefully hear from me, as many of you may have read about SAGE and their postpartum depression drug, you'll hear from our colleagues at Compass. I think we all agree that these are very complex conditions, but we are making a lot of headway, and there's reasons to believe we can make even more headway. So I think in any disease, you need to basically look at things at this level. You need to understand things at a genetic level, then understand how that affects a protein network, cell function, and then the complexity really starts for us. Ultimately, we're not talking about conditions that arise out of problem from single cells, but rather integrated synapses and circuits. And ultimately, those circuits lead to these complex symptoms we have: mood, cognition, psychosis, anxiety, anhedonia, etc. And so we've got to make sense of all those things. I think, as you heard from Kerry Ressler, a lot of these conditions are very heritable, very genetic. But one of the problems is that these are conditions that arise out of the inheritance of multiple genes, probably hundreds, of small effect. And of course, as we know right now with COVID, and there's big environmental effects. So we've taken the view that rather than trying to study things at a fundamental single gene by gene, let's see where these things come together, namely at the level of synapses and circuits, and then see if we can make a difference there. One of the reasons for taking this approach is that the last 15 years or so, elegant work from Rick Hugonero, Roberto Malinau, Rob Malenka, David Brett has really identified some of the molecules involved in synaptic plasticity. And you can think about many definitions. One definition is the way in which you regulate the strength of information flow through specific synapses. And their work tended to show that the movement, sort of shown on this cartoon on the left, the movement of certain glutamate receptor subunits, AMPA or NMDA, into or out of synapses really played a critical role in regulating the strength of synaptic function. And so while I was with Tom at the NIH and under his leadership as NIMH director, we did a lot of basic science work to look at whether these plasticity molecules, AMPA and NMDA, could play a role in depressive phenotype, and could targeting them lead to the development of novel therapeutics. We argued that they were arguably the correct targets. And so if you hit them directly, you might have treatments that work rapidly. And we argued that you might even have treatments that worked when other treatments didn't work, for example, SSRIs, because SSRIs basically start very much upstream, increasing serotonin levels in the synapse that then goes through a cascade of events to regulate these plasticity molecules. And I think, as most of you know, Dennis Charney and John Crystal at Yale had done some studies showing that low doses of IV ketamine and NMDA antagonists had antidepressant effect. So while we were at the NIH with Tom and Carlos Zarate and others, we undertook this trial where we brought in treatment-resistant depressed patients. They had been depressed continually for 3 years. Many of them had failed more than 6 treatments. They were given either IV low-dose ketamine or low-dose placebo. And we were struck to find that in this highly refractory population, within 2 hours, we started to see an antidepressant effect. At 24 hours, 70% responded. And if you look at the green bars, I need to point out that that's obviously from a different study. That's from the STAR-D study. But there you see that the available existing antidepressants at 8 weeks give you something like a 15 to 20% response rate. So these were obviously very exciting results. I left NIH about that time. When I first came to J&J, the plan wasn't to necessarily, focus on ketamine's findings. Rather, we were focusing on some of the other molecules, but then started to think of, if you've got these remarkable results, can we develop an FDA-approved, safe, effective treatment? My hypothesis was that the rapidity of antidepressant response, in addition to being because of the right target, you are getting the target engaged very rapidly. And I postulated that you could achieve that with a nasal administration as well. Now, anyone who's taken a nasal decongestant knows you're delivering a very small amount of drug. So we initially did studies to show that the S version of ketamine was 3 times more potent at the NMDA receptor, which would suggest that you could get away with 1/3 the dose of S-ketamine as you could regular ketamine. So on the left of the slide, the first studies were to show that S-ketamine, that's IV S-ketamine, actually worked. And as you can see, almost as predicted, 0.2 milligrams per kilogram of S-ketamine had this robust and rapid antidepressant effect, which is what's generally seen with 0.5 milligrams of regular ketamine. Having established that, we then moved to the middle panel, which was to figure out how do we deliver an intranasal dose that recapitulates some of those findings. And then on the right-hand side, then deciding, okay, how do we treat people? Because traditionally in medicine, you sort of think you've got to have round-the-clock receptor occupancy, 24/7 receptor occupancy. Here we postulated that if you're hitting these plasticity pathways, you can actually work intermittently. So we developed a paradigm where for the first 4 weeks, you get the drug twice a week for the first 4 weeks. And then once you're well, the treatment gets spaced out until basically depending on what you need, once a week, once every 2 weeks, once every 3 weeks. And what's shown at the bottom of the slide is sometimes people wonder why it takes, so long. Not only do 95% of things fail, but we had to do 19 phase 1 studies, 4 phase 2 studies, and 6 phase 3 studies. Here's some of the data. So on the left, you see the results in treatment-resistant depression. You see that about 70% of patients responded and about 50% of patients went into remission. On the right, once again, you're looking at the results from the STAR-D study, and you can see at 8 weeks, you see a markedly lower response or remission rate compared to what we saw with Spravato. And what's noted at the bottom is that the majority of the effects were actually seen within the first 24 hours. Obviously, we know that depression is a chronic condition, and so you not only need to get people better, you need to keep them better. And once again, the data from the STAR-D trials are instructive, where they found that even once you got people well, about 70% of those people had relapsed despite being maintained on the medication after 9 months. So when we did our studies, we found almost the inverse of that, where about 70% of people remained well at 9 months, suggesting that this is certainly a treatment that gets people well rapidly, but with repeated intermittent administration, you can also keep them well. The rapidity of onset also comes to the question of, seriously ill patients. And it may surprise people to know that by and large, every trial of an antidepressant has actually excluded suicidal patients because, no one wants a suicide to take place during a clinical trial. Thanks to the leadership of my colleague, Carla Canuso, we actually did the opposite, where we went to look for people who had not only suicidal ideation, but suicidal intent, either in the emergency rooms or in inpatient units. Then we recruited them, obviously, with informed consent into the trial. Given that this was such a sick, ill population, we wanted to give them the best standard of care. So they were basically hospitalized, given support, given therapy, other medication was started. And then on top of that, they got either placebo or either intranasal ketamine. And on the right, you see two big findings. One is that environmental effects matter. So when you hospitalize people, when you provide them with support, when you take them out of distressing situation, you can see they do see some relief of their depression. But on top of that, we saw the marked antidepressant effect of Spravato, and we were delighted some months ago to get the approval for this second indication. Our hypothesis, as I mentioned, was that this was occurring by complex regulation of NMDA and AMP receptors. And what's shown on the left is how we think NMDA antagonists work. So they're thought to exert their effects on GABAergic interneurons, which disinhibits these glutamate neurons. So you get a burst of glutamate release that then activates AMP receptors. So if that's correct, then if you block AMP receptors, you should theoretically block the antidepressant effects of an NMDA antagonist, even though the NMDA antagonist isn't working on the AMP receptor. If the effects are going through AMP receptors, if you block the AMP receptors, you'll block the antidepressant effect. And that's what we see in the middle panel. In panel B, you see, these rodent depression models, when you pre-treat them with an AMP antagonist called MBQX, you don't see anything, but it completely blocks the effects of ketamine. And in panel C, considerable data suggests that the NR2B subunit may be particularly responsible for antidepressant effects. So when you use a selective NR2B antagonist, once again, you see antidepressant effects that are blocked by AMPA pre-treatment. Now, you can't You can't stick electrodes in human brain and measure AMP currents, but there's strong reason to believe that a lot of the excitability you can measure through things like magnetoencephalography is mediated by AMP currents. And so we did the study on the right. We were able to show that indeed the somatosensory cortex excitability was highly related to the antidepressant effects. All of which suggests that AMPA receptors may also be a very relevant target. One of the problems is that AMPA receptors are almost too important. They're present throughout the brain and they regulate so many functions, so you can't just hit them with a sledgehammer. Again, important advances in biology identify these accessory proteins shown on the left called TARPs, and our own David Brett did a lot of the work. And what he was able to show that these accessory proteins are present in a very regionally selective way. So if you look in the middle panel, for example, if you look at the right, the red circle, that's one of these accessory proteins called stargazin, which is present almost exclusively in the cerebellum. By contrast, if you look at the green circle in the bottom, that's another accessory protein called gamma-8, and that's present almost exclusively in hippocampal and limbic regions of the brain. And then what that translates into is that if you develop a drug working on the gamma-8 TARP, you have no effect in areas of the brain where this receptor is not expressed. For example, if you look at the tracing in B, you can see that in the cerebellum region, you see no effect. By contrast, if you look at the tracing in C, you can see that you have a clear effect on the tracing there. But once again, it's an attenuation, not a sledgehammer. Now, on the right, what you see is something we feel very important about. A lot of drugs have failed because you haven't even really known exactly how much of the drug is occupying the receptor in a specific brain region. So we developed these things called PET ligands for all our drugs. And here you see the AMPA gamma-8 ligand, and that allows us to pick out the dose, to, for example, to occupy 50% of it. And now we've got clinical trials underway. I think, as Leanne eloquently pointed out, is that we really need to move beyond the sort of one-size-fits-all to recognizing both subtypes of depression are also domains within this depressive phenotype. I think most of you know, if you go to the DSM diagnosis of depression, you could have two people meeting the criteria for major depression with almost no symptoms overlapping. And obviously that just tells you that this is not a single condition. So we've been very interested in mapping some of these key symptoms onto neuronal circuits and then targeting those circuits to bring about improvements in those symptom domains. And one of those relates to the orexin circuit. And there's reason to believe that the orexin circuit, and in particular the orexin 2 receptors, really play an important role in regulating what we call hyperarousal. That manifests as ruminations, insomnia, people being wired. So we've argued that if you have an orexin 2 antagonist, you would arguably really help people whose depression is characterized by those symptoms. And that's what you see here. Now, one of the problems is that we know it's not really a problem. Clinically, it's beneficial, but orexin 2 antagonists help sleep. And as you know, if you look at the Hamilton Depression Scale, sleep is a big item. So how do you show that your effects aren't just due to helping people sleep? And what you see on the left is the adjusted Hamilton Depression Rating Scale, where the insomnia items have been removed. And on the right, the 6 core symptoms of depression that don't include insomnia. And you can see that the orexin 2 antagonist is having an effect on the core symptoms of depression. And now we're in phase 3 trials to look at, arguably subpopulations of depressed patients whose depression is characterized by this hyperarousal that might respond preferentially to this treatment. And similar efforts are underway with other molecules targeting patients who have prominent anhedonia symptoms or patients who have very prominent anxiety, or sometimes called anxious depression. I think one of the other things that you've heard about is that depression is associated with a lot of medical comorbidity. So cardiovascular disease, type 2 diabetes, both pre and postmenopausal women have high rates of osteoporosis. And this is one of the reasons why people have argued and basically measured different inflammatory mediators. And lots of studies, some better studies, some not so good studies, have shown that depression is associated with an elevation in certain pro-inflammatory cytokines. What you see on the right here is, one particular cytokine, IL-1 beta, and you can see that either bipolar depressed patients or unipolar depressed patients have these elevations. And that seemed to sort of make sense. Aha, this is why patients with depression, in addition to lifestyle reasons, have these, medical comorbidities. But many investigators, most notably Ranga Krishnan when he was at Duke, wondered whether there might be a subpopulation of depressed patients in whom the inflammatory mediators could even be driving the depression. And I think, like everything, it's not a non-specific brain inflammation. There are specific inflammatory molecules that have big neuronal effects. And what you see on the top there is this work by Rob Malenka showing you the effects of, for example, TNF-alpha on very specific plasticity molecules and basically electrophysiological readouts of plasticity. Some years ago, basically because a lot of companies were leaving neuroscience, the Wellcome Trust in the UK contacted me and asked if they could have a several-day event showcasing British neuroscience, and we were delighted to participate in that. And one of the things that came out of it was a neuroimmunology of mood and Alzheimer's consortium. And what you see at the bottom are some of the results from that consortium. So Gail Wittenberg and our group analyzed the data from multiple clinical trials of immune modulators targeting TNF-alpha, IL-12/23, IL-6, and so on, and asked the question, gleaning readouts of depression from other rating scales, does your treatment with these modalities improve your depression? And if you look on the left, you can see that 3 of those are basically to the right of the dotted line, namely TNF-alpha, IL-12/23, IL-6, which suggests that people's depression was getting better under treatment. And what everyone's thinking is, well, of course your depression got better if your rheumatoid arthritis got better or your lupus got better. So you look in panel C there, and that's looking at people whose primary immune condition did not respond. And you see that their depression still did respond. So many people are now trying to use monoclonal antibodies to tie up these cytokines in the periphery with the idea then less will get into the brain. We've taken an alternate approach. So there are these cells in the brain called microglia, which are the immune cells in the brain. And one of the key regulators of microglial function is something called the P2X7 receptor. And we've developed a selective P2X7 antagonist, which really regulates microglial function. And as you can see, again, on the right, we've developed a PET ligand. And now, having done that, we're currently in trials for major depression as well as bipolar disorder. And this is coming back to what Leanne was suggesting as well. It's unlikely that this is going to treat all patients with depression, but it may be a subgroup of depressed patients whose depression is driven by some of these immune mediators. I'm not talking about autism, but autism spectrum disorders, it's really one of those almost classical conditions where the genetics and neurobiology has really focused efforts on the synapse. The slide shows many of the targets. And in principle, going after so many of these targets should help, but we don't have a treatment yet. And we think that one of the problems has been that the clinical trials have been so challenging given the heterogeneity. So there's many types of heterogeneity, and we've developed these tools and technologies, something we're calling Janssen Autism Knowledge Engine. The first thing is on the left here, something called MyJake, which is basically recognizing that there isn't an autism doctor. You have to gather information from the person themselves, their parent, pediatrician, school teacher, etc. And so we've developed on the left this MyJake to basically help document and collate symptoms from all these informants. Then in the middle is, again, something Leanne was talking about, as much as you can come up with objective measures rather than, asking people how you're doing. And as we know, there's a number of biological abnormalities, things like eye, gaze, autonomic symptoms, etc. So Jake Sense is a platform that picks up these different biomarkers and quantitates them. And then on the right is something called Jake Stream, where we can then identify subgroups of people who have specific biomarker changes and specific clinical symptom domains that we can then target with specific treatments. And we now have a proof of concept trial underway. And I'm using that as a segue to what I think is also going to be very powerful, namely the digital technology advances, because our diseases and disorders are characterized by multiple recurrences and relapses. And right now we sort of wait until people get worse and then show up in the hospital or the emergency room. Wouldn't it be fantastic if you could get some early warning signals about relapse and depression, switch into mania, worsening suicidality? And I think if done correctly, some of these digital technologies can help us there. And what's shown here is the ability to identify depressive relapse weeks before clinical diagnosis using different sensors and smartphones. And I'm sure most people know voice and speech are turning out to be very powerful. Obviously, we need to be very thoughtful about privacy, about data ownership. Our thinking is that the person should own their own data and then give permission to, should they be alerted if there's a deviation, their loved one, their treating team, etc. But if we do this correctly, this could really help us make a difference for our patient. And so to conclude, and with this call for action, I think it's incumbent upon all of us to help society understand that these conditions really, unfortunately, are highly disabling, devastating, and goes without saying, affect every facet of people's lives. I think I'd like to leave with a message of hope that while we have a long way to go, we are making a lot of progress. And as Tom alluded to, we need to come together as a society to tackle stigma, raise awareness. But I also think that the scientific advances, much like we've seen in HIV/AIDS or in cancer, will help with the stigma. I think we need to get to this idea of preemption, so move from diagnose and treat to predict and preempt, so we can preempt the ravages of the illness. I think as Tom alluded to, and I think Brandon mentioned so eloquently with the Aspire program, there's no doubt that these are biological conditions, but to get optimal outcome, you need to think more holistically about people. So marry the biological intervention with different kinds of services. And as I said earlier, I think it's a shared societal responsibility. Many of us, including Tom, including Brandon and myself, are delighted to join forces with Garen Staglin, Victor Zhao, and others in launching this Healthy Brains Global Initiative, which is trying to bring $10 billion of new research for brain health, starting with youth mental health. And for those of you who are interested, that website will give you more information. I'll stop here and take any questions you may have. Thank you very much.
Well, thank you, Husseini, as always. That was a spectacular overview of so much work. We heard about NMDA, AMP, orexins. You talked about cytokines and immune factors, and then digital on top of all that, all in 20 minutes. We don't have time for a lot of questions. A bunch of questions have come in, but I can't let you go until I ask one that I'd love to get your vision on. If you put your comments, Leanne's comments, and everything else you know about what's happening together, just give us a sense of how you look at depression treatment in 2030. What's that going to look like in 20 seconds, if you could?
Yeah, I think it's definitely going to be along the lines of what she suggested, that it's not going to be a one size fits all. We're going to tailor treatment to people. I think sometimes people think that, would a big pharma actually want that? Aren't you actually narrowing your market? But I think everyone actually recognizes if you can treat a subpopulation really well and they stay on your treatment, it's actually much better than starting with, an antidepressant that you try and fail, etc. I think, again, with our healthcare crisis, society is demanding evidence. And so that the more evidence we can generate to show that we're actually giving people their lives back, you're not just, treating symptoms of depression a little bit, but you're allowing people get back to work, to schools, and function in society. I think that'll be very important to show the value, and hopefully we can get psychiatry to be viewed as important a subspecialty as many of our other subspecialties. Thank you.
Terrific. Hey, Husseini, thank you so much for being part of this today. And it's really, as always, it's an inspiration to see all the things that you're working on and thinking about. So good luck with all of that.